Welcome everyone. This is our first lesson for remote learning living environment. We're happy that you guys are here and tuning in, and we're really excited for this new learning platform. So, if you watched our welcome video, we encourage you guys to be sitting at a table or a desk and that you have a notebook or paper and something to write with. And we will tell you throughout the lesson where you should be taking notes and where there will be check for understanding questions. So if you need to pause the video and get settled, you can do that now. And we are going to start today, like we start every class, with our mindful minute. So we'll do our deep breathing that we all know how to do for 1 minute. So wherever you are, go ahead and sit with a mindful body. Make sure your head, neck, and spine are aligned. And we'll take deep breaths in and out through our nose for 1 minute. So get ready. And together, we will take a nice deep breath. Inhale through your nose. And exhale. Inhale through your nose. And exhale. Nice deep breath in. Exhale. Inhale deeply. Exhale, pushing out the air. We're halfway there. Inhale through your nose. And exhale. Inhale. Exhale. And last one, nice deep breath in. And as you exhale, push the air out and release any tension. Allow your breathing to return to normal. Check in with yourself internally. Notice how you feel, if anything has changed, if there's any new emotions or sensations, and maybe not. And that's okay. When you're ready, you can slowly open your eyes if they were closed. And let's get started. So this is week 1, lesson 1, DNA, replication, and transcription. Now we have posted the lesson from the last week we were in school. So from March 11th and 12th, we started this unit and we posted that lesson. So if you were absent or if you need a refresher, we encourage you to go back and look at the slides for the lesson before this one. So when you see this little icon over here, this means that you should be writing this down. So having your pen and pencil, you're going to do the do now with us. We're going to do it all together. So 1, recall the base pairing rule for DNA. And 2, using that rule, create a new strand of DNA for the following template. Now, this whole unit is genetics, so that's why we're talking about DNA. If you remember the base pairing rule, it looks something like this. So here's your strand of DNA, and we have our nucleotide bases, which are A, T, C, and G, which stand for adenine, thymine, cytosine, and guanine. The base pairing rule is what letter pairs with each other. So A goes with T and C goes with G. So using that rule, what would be our new strand of DNA? So the A would pair with T, T would pair with A. C pairs with G. G pairs with C. A goes with T. C with G. G with C. And C with G. So that is our base pairing rule for DNA. All right, excellent. Now, how do I erase that? All right, so again, we're seeing this icon over here at the top right corner, which means you should be writing this down. So today's learning target is I can explain DNA replication and transcription. If you need to pause the video to write that down, you can do that now. So DNA replication and transcription are two processes that DNA can go through. Now, why is DNA so important? If you remember from our last lesson, DNA holds the instruction or the code to make proteins. And when we think of proteins, oftentimes we think of the proteins that we eat in food or protein in like chicken. However, there are thousands of different kinds of proteins and they have all different kinds of functions or jobs. So over here is a website that links to, or a link that goes to this website. You're going to open this in your phone. And it has all these different categories of different types of proteins, which is super cool. So for example, I'm going to click on this snake one because this is interesting looking. Tells us about sensory proteins. So sensory proteins help us learn about our environment. They help us detect light, sound, touch, smell, taste, pain, and heat. Some organisms can even detect electricity or magnetism. So if we're looking at our snake over here, it says the TRPA1 protein in a rattlesnake's pit organ senses body heat. This sense helps the snake locate its prey. So maybe you knew that snakes can sense body heat, but did you know that it was caused by a protein? Super cool stuff. You can feel free to read through all of these different kinds of proteins. If we go back to our lesson, if you see this little purple piece of paper icon thing, this shows us or tells us that there's something you need to do on your Google form. So you're watching your lesson and there's all these questions and check for understandings down here. So this signals for us that we need to go to our Google form and do something. Now the first question here says, visit the types of proteins website, the website we were just on, and type in one protein and briefly describe its job. So for example, I would write TRPA1 protein in snakes senses body heat. Whoa. So you can do this as you watch, or you can answer all of these questions after. That's up to you. But this is just to let you know that there is a part on the Google form that goes with this part of the lesson. All right. So DNA can do all of these things. It can turn into RNA, which eventually turns into a protein, and it can also do something called replication. So if you hear the word replicate, what does that make you think of? Well, replicate means to make a copy. Sorry, this down here says DNA copies itself. So DNA can make a copy of itself. DNA replication is exactly that. If we look at our picture here, we have a mother cell going through something called cell division, and then what happens? it splits into two cells. So this cell divided, and now there are two identical copies of this cell. So DNA replication is making an identical copy of DNA for cell division. Now again, we see our icon over here, which means we should be writing this in our notes. Pause the video now so that you can go ahead and copy down that definition. All right, so why would our cells need to make copies of themselves? This is a question that shows up on our Google form. So again, you can answer that now or you can answer it later at the end of the video. So why do our cells need to make copies of themselves? Well, we grow and different parts of our body grows at different times. So if you look at this picture, your hair grows 0.35 millimeters every day. Your red blood cells are replaced every 120 days. Your liver cells are replaced every 300 to 500 days. Heart muscle cells actually stop growing when you're about 10 years old, and your visual cortex also stops growing. So there are parts of your body that are complete with their growth stages, but there are many parts that need to continue growing. So your cells need to make copies of themselves in order to allow your body to grow. Also, if you think about it, think about what you looked like when you were little. That's Beyonce, FYI. And think about what you look like now. You probably do look like Beyonce because you're a superstar. And you've grown so much from when you were younger. And that is possible through cell division. Why else do cells need to make copies of themselves? Well, your body needs to repair itself. So whenever you get an injury, you get a cut, your body needs to heal and repair. Another reason that cells make copies of themselves is for something called asexual reproduction, which we'll talk about in a later day. But this is mostly used, or an example that we're using here is like a starfish. If you cut off an arm of a starfish, it can actually grow its arm back. And that's done through cell division. So how does DNA replication work? We're going to watch a video. Again, there are questions that go with this video. And you're going to want to take, if anything pops out to you, you can take notes on that as well.
It might be hard to believe, but at the very start of your life, you are a single, microscopic cell called a zygote.
Sorry, I'm going to pause and put the captions on, and I'll just restart.
It might be hard to believe, but at the very start of your life, you are a single, microscopic cell called a zygote. Your body now contains millions of cells, which all came about by the process of cell division. Cell division happens when one cell divides to form two cells, and it is the basis of growth and repair in humans. It's also used for asexual reproduction in organisms like bacteria. But a single cell cannot just split itself in half to form two whole new cells. They would essentially only be half a cell. Before the cell divides, everything inside needs to be copied. This includes all of the parts of the cell, like the mitochondria, as well as the chromosomes inside the nucleus. A human body cell contains 23 pairs of chromosomes, 46 altogether. During cell division, the DNA in the chromosomes is copied or replicated to form 46 pairs, double the number of chromosomes. These pairs split apart when the cell divides to form two new daughter cells, each having the correct number of chromosomes. The splitting apart of the chromosome pairs happens during a stage in cell division called mitosis. Let's take a closer look at DNA replication. DNA is made up of many nucleotides, each containing a base represented by the letters A, T, C, and G. A piece of DNA contains two strands of nucleotides, twisted together to form a double helix. The strands are complementary. This means that whenever there is an A in one strand, it will be joined to a T in the opposite strand, and wherever there is a C, it will be joined to a G. If you are not familiar with the structure of DNA, then take a look at this video. During DNA replication, the double helix unwinds. Then, an enzyme called DNA helicase unzips the DNA so the two strands are separated. This happens at several points along the DNA. An enzyme called DNA polymerase attaches itself to the DNA strands and is used to add complementary free nucleotides to the now exposed bases on both strands. A pairs with T and C pairs with G. This forms two DNA molecules, each of which have one brand new strand and one from the original DNA. These two strands twist to form a double helix. The two new DNA molecules are both identical to the.
All right. So, boop, boop, boop. So as you saw in the video, DNA will replicate itself by becoming unwound. So if you look at this picture here, it comes undone. And then there is a special protein that comes and adds on the bases with using the base pairing rule. And at the end, there are two identical strands. So during DNA replication, your DNA becomes unzipped, and then a protein called helicase comes and separates it, and you have these two strands. Then another protein comes and it attaches new base pairs to the old strands using the base pairing rule. And then at the end, you have two identical strands of DNA. So that's how DNA will replicate or make a copy of itself. So, how does DNA actually make things like proteins? So, all those cool proteins that we saw, how does your DNA actually do this? This is done through two processes called transcription and translation. This is separate from DNA replication. So, DNA replication was for copying your cells being able to divide. This transcription and translation process is for DNA being able to make proteins. So here was DNA replication. Now we're moving on to transcription. What does transcribe mean? Transcribe means to take information and put it into written form. So we'll see DNA being used as information and being written as RNA. And DNA is going to act as the template, which means the guide for when RNA is made. And later, we're going to look at how that becomes a protein. So right now, we're just focusing on transcription. So what is RNA? You can think of RNA as like the cousin of DNA. So it's another nucleic acid, but there are some differences that we need to know. So if you look at this picture, what do you notice right away? You'll see that RNA looks kind of different, right? RNA only has one strand, whereas DNA has two strands, which make the double helix. What else do you notice? The bases on RNA are a little different. So they both have cytosine, C, they both have guanine, they both have adenine, but RNA has something called uracil, while DNA has a base called thymine. So we're going to watch a video comparing DNA and RNA, and you can take notes on this, and there is, again, a check for understanding over here. So in your notebook, you can make a T chart that looks like this with DNA on one side and RNA on the other side, and you're going to write down differences between the two as you watch the video. If you need to pause to set up your notebook, you can do that now.
Let's talk about the differences between DNA and RNA. So DNA is usually double-stranded, has two separate strands that are connected, which look like this, while RNA is single-stranded most of the time, looking like this. DNA is much larger than RNA, with usually millions of nucleotides. And RNA is, of course, smaller, with hundreds to thousands of nucleotides, and sometimes tens of thousands. DNA has a deoxyribose sugar, that's why it's called deoxyribonucleic acid. Its sugar structure looks like this. And in DNA, of course, it has the phosphate group and the nitrogenous base. And this carbon here is missing the oxygen. As you'll see with our RNA, which has a ribose sugar, looking like this, again with the phosphate group and the nitrogenous base. And this carbon here, the number 2 carbon, has an oxygen attached to it.
So this part right here where they're talking about the structure of the sugar, you don't need to know this, but this is where DNA and RNA get their name from. So DNA stands for deoxyribonucleic acid, and RNA is just ribonucleic acid. So again, this is you don't need to know these structures, but that is one of the differences. That DNA has this sugar called deoxyribose sugar, and RNA has this sugar, which is called ribose sugar.
Oxygen attached to it. The location within the cell for DNA is primarily located in the nucleus, with some DNA being in mitochondria as well. For RNA, while some is in the nucleus, most of your RNA is in the cytosol and in the ribosomes throughout the cytosol.
Cytosol is another term referring to the cytoplasm. So if this is our cell here, our nucleus is this organelle, and our cytoplasm is the jelly-like substance surrounding all of the organelles. So DNA is found in the nucleus, RNA is mostly in the cytoplasm and ribosomes, which are not pictured here.
For your nitrogenous bases, DNA has 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. And RNA also has 4 bases: adenine, guanine, cytosine, and uracil. So RNA does not have uracil, does not have thymine, but has uracil instead.
Okay, so hopefully you were able to write down some differences between DNA and RNA. You can always pause this video and go back if you need to. You can take a moment to answer the check for understanding questions, or again, you can do that at the end. So, DNA is used as the template or guide for RNA. So, that is our process of transcription. Here's a definition that you should write down. Transcription is the first step of protein synthesis and happens in the nucleus. So remember from the video that DNA is mostly found in the nucleus. This is also where transcription happens. So transcription uses DNA as the template or guide. And if we're thinking of the word transcription, That means to put information into written form. So here we have mRNA, that's a type of RNA, which is being written based off of the information of the DNA. So what does that look like? Looks like this. It's very similar to when we did DNA replication. However, what do you notice about these pairings or the pairing rule? That's right. mRNA will not have T and instead it has U for uracil. So your base pairing rule for DNA and mRNA would have U pairing with A. There is no T in mRNA. So here are bases just to review. Again, mRNA has uracil instead of T. So let's look at what transcription looks like. This is a really cool animation showing what it looks like inside the cell.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
So right here is your mRNA starting to be made or starting to be transcribed. So here, this whole structure is our DNA. It became unzipped, just like in replication, but now the one strand of DNA is serving as the template or guide for the new mRNA. How do we know this is mRNA that's being made on the top? We know this is mRNA because it has this U base for uracil. And if you look, there are no T's, it's just U.
The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Moves along the DNA.
We're just going to watch that again so you get another visual of transcription.
Switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
So that's the new mRNA coming off of the DNA, which served as a template or a guide. So just to review, DNA, we looked at two processes that DNA can go through: replication and transcription. DNA replication is when DNA makes a copy of itself to prepare for cell division. And this is used for processes like growth, repair, and asexual reproduction. And transcription is when DNA makes RNA or mRNA, and it's the first step of protein synthesis. And what is protein synthesis? Proteins are important for us. They serve many functions like eye color and texture of hair, skin color, many, many functions. So replication means to copy, and transcription or transcribe means to put information into written form. That's one way you can remember it. Replication is when DNA makes a copy, and transcription, if that means to write, we can remember that mRNA gets written from the information of DNA. So now that's the end of lesson 1. You can go back to your Google form and answer the check for understanding questions. And then after that, you will go to assignment 1 and you'll see that these are practice questions based off of lesson 1. Okay, and make sure you hit submit at the end. You'll get a grade. You'll see how you're doing. Okay, so thank you so much for watching, for learning, and we will see you in lesson 2.